Hi guys, welcome to Diffusion and Osmosis. So in terms of the specification, we already were looking at the structure of the cell membrane. Now we will be looking at the simple diffusion, facilitated diffusion and osmosis. So the keywords associated with this will be still phospholipid bilayer, will be carrier and channel proteins, okay, and we will be looking at the water potential for osmosis. So those are the different types of the transporter you need to know for your specification. And for today's topic, we will be looking at the simple diffusion, facilitated diffusion, and osmosis. All those three are examples of the passive transport, and they do not require any ATP. So what is then diffusion? Simple diffusion is the net movement of the molecules down the concentration gradient. So that's really important. You get used to the terminology with the concentration gradient. So down the concentration gradient, and if at any point you've forgotten and you want to write this down, you can possibly get away with saying from higher, but look, ER to lower, okay, concentration, but make sure down the concentration gradient is the one that you will use. Diffusion, as we've mentioned, is a passive process, doesn't require any ATP. So what can actually diffuse through the phospholipid bilayer? Small, nonpolar, and lipid-soluble molecules can pass through the phospholipid bilayer. So the rate of diffusion will depend obviously on many things. So the concentration gradient, because we've said from higher to lower concentration, a distance over which diffusion takes place, area over which diffusion takes place, the size and nature of the diffusing molecule, and the size of the pores over which diffusion takes place, and obviously the temperature, because with the temperature will be more kinetic energy, okay, so faster diffusion. And membranes are hydrophobic, so uncharged nonpolar molecules and lipid-soluble molecules can diffuse through the simple diffusion, of course, through the membrane, what we've mentioned before. So then facilitated diffusion, it's here to help with charged ions, polar molecules, okay, which cannot just pass easily through the phospholipid bilayer. So hence we've got the proteins to help out. So the proteins that will be involved in facilitated diffusion are channel and carrier proteins. This diffusion still takes place down the concentration gradient and does not need any ATP. So it's a passive process, obviously depends on the kinetic energy here, because more kinetic energy, obviously faster diffusion. We don't need any ATP, occurs down the concentration gradient, which we've mentioned, and there are specific proteins. What we need to remember, one protein for one thing, okay, hence specific. So what is actually the difference between those proteins? So channel proteins are for water-soluble molecules, and some channels are open all the time, but others open when a carbon is attached, for example. And the carrier proteins, those proteins change the shape when the molecule binds with them, allowing the molecule to diffuse through. So this is the channel proteins, as you can see a little corridor here, so that's a channel, and carrier proteins will pick on one molecule from the other side, change the shape and release it on the other side. So describe how substances move across cell surface membrane by facilitated diffusion for 3 marks. So let's see if you can recall our keywords now. So carrier or channel proteins are involved. We say specific, one for each thing, complementary to the substance that they're going to carry, and down concentration gradient. So look, not such a thing like from higher to lower. Get used to the new terminology. Right, so again, key terminology, give one property of the molecule X, okay, that can diffuse, okay. As you can see, what kind of diffusion is it? Yes, it's a simple diffusion because it goes through the phospholipid bilayer. So what did we say? Small, non-charged, and lipid-soluble molecule, okay. So another thing here, we've got the rate of diffusion and concentration. Okay, so we need to look at the limiting factors here. Limiting factor is the concentration of sodium ions. Where this is coming from the fact that affect the excuse me, affects the rate of diffusion. Okay, because that concentration will affect the concentration gradient. Okay, as more concentration, okay, it's the variable, independent variable that increases, so does the rate of diffusion increases. And explain the shape of curve between C and D. Right, what's going here? Firstly, before you explain, you need to describe in your head. So what can you see? Levels of Y. So referring to what we've just said here, you need to look at the concentration. So the sodium ions are passing through the channels at the maximum rate, and the rate is limited by the number of sodium channels. Another limiting factor here, because concentration increases but the rate levels off. Okay, so here what could be another limiting factor are the channel proteins, because it's a facilitated diffusion. So osmosis, then another key terminology, we need to say that osmosis takes place, okay, from higher water potential to lower water potential. Okay, so that's another terminology you need to be aware of. So few things. What's a solute? Could be, for example, glucose, so the substance that it's dissolved in solvent, for example, water. So solute and solvent makes the solution. Okay, and key things about water potential. Water potential, it's a pressure created by water, and it's measured in kilopascals. The highest water potential is 0. So any other values will be obviously negative. So if the values get more negative, okay, so 0 the highest, -1, -100, okay, -100, okay, has the lower water potential because it's more negative. Okay, and what we need to remember, that adding solutes makes it more concentrated, makes the water potential lower. So what does that mean? It's more negative. Okay, so less water, more solutes. So here we've got the situations. What happens to the cells? So there is no change. What that means, that the water potential outside and inside the cell is the same. Okay, so net movement is 0. Here the cells was rest. So that means that the water comes in. And anytime you talk about osmosis, you need to say where is higher water potential. So movement of water from higher to lower water potential. So higher water potential outside, lower inside, hence the direction of the movement. Here the cell shrinks. So what happens? Okay, at the beginning here must be higher water potential, lower in here. So the water leaves the cell by osmosis from higher water potential inside to lower water potential outside the cell. Right, so similarities between diffusion and osmosis. So movement down the concentration gradient, what we say, both passive, okay, don't need energy. That's everything for the osmosis, facilitated diffusion and simple diffusion. See you later.